Hi everyone, I'm Sarah, and today we'll be talking a little bit about synaptic transmission, which is essentially looking at how neurons communicate with each other. And so, basically, how synaptic transmission works is you have a neuron that sends a signal. So, this is neuron A. We usually call it the presynaptic neuron. And then we have a gap between the neuron to the next neuron, and neuron B is on the other side of this gap, which is called the postsynaptic neuron. And you can guess kind of that the space in the middle is called the synaptic cleft. And this space between the neurons is sort of just the extracellular space between cells. And this is about, you can see about 20 to 30 nanometers, so really, really small. And before I go on to sort of explain a little bit more about synaptic transmission, one thing to think about with neurons, so neurons, they communicate through two methods of communication: electrical communication and chemical communication. And for this purpose of synaptic transmission, that is under chemical communication. We'll talk about electrical communication on another video, but synaptic transmission comes under chemical communication. Chemical communication, as you can guess, involves the use of chemicals that are released from the presynaptic neuron and travel to the postsynaptic neuron across the synaptic cleft. And so the way that these neurons travel is that you'll have some electricity that travels to this neuron A. This electricity is part of the electrical communication. This electricity causes a wave of positive ions to come into the cell, into this end of the cell right here. And this wave of electrical, this positive ion causes the release of these vesicles, and these vesicles are just pretty much big bubbles that I like to call them, essentially, that carry chemicals. And these chemicals, the name of these chemicals are called neurotransmitters. And what's cool about neurotransmitters is that they have the ability to fuse with the membrane of the cell right here. So you can see the membrane, very thin. I mean, in reality, it's a lot bigger and actually thicker, but the membrane, the vesicle fuses with the membrane, and it causes the release of these neurotransmitters into the space. And some of them travel to the other side and can bind to receptors that are embedded on the postsynaptic neuron, which triggers a whole bunch of things happening, and eventually the signal will continue on this neuron. Now, for sake of simplicity for this video, I'm just going to be talking about the presynaptic side. We'll talk about the postsynaptic side in another video. But really with the presynaptic side, we're going to really zoom in on what really is happening, first about this fusion process, and then going into a little bit more detail about how exactly that these vesicles actually begin to release these neurotransmitters to begin with. And so starting with, again, I mentioned this wave of positive ions. So that's the start of our synaptic transmission pathway. So I'm going to zoom in a little bit. There are positive ions, and we're going to zoom in on this particular spot in the plasma membrane right here. Plasma membrane, and we can ignore the rest for now, but the plasma membrane has something called voltage-gated calcium channels. So these are calcium channels that have the ability to sense changes in voltage. So I'll write that down right here. And these voltage-gated calcium channels sense this positive rush of ions from the electricity that's coming through from this neuron, and they sense this and they allow calcium to diffuse into the neuron right here. So they diffuse in, so the calcium ions coming in. Now the calcium ions comes in because you'll see that outside of the neuron in the synaptic cleft, out here you'll have a high concentration of calcium. So a lot of calcium out here, a lot of calcium. We'll zoom in a little bit here. A lot of calcium, a lot of calcium. It comes in, diffuses in, so I should actually erase this first. Diffuses in a lot of calcium, not much calcium inside the neuron, so it diffuses in. You'll bring more calcium inside the neuron. Now, what happens inside the neuron is that these voltage-gated calcium channels are usually located right next to proteins called docking proteins on the plasma membrane. And they help facilitate that docking that usually happens for the vesicles. And we'll zoom in actually even more now in our second picture right here, where you can see this vesicle that I've drawn here, outlined in blue, with a bunch of neurotransmitters inside. And these neurotransmitters are still inside, and you can see that, so outside here is the synaptic cleft. So we need the chemicals to go from inside the neuron, inside this vesicle, to go to outside the neuron. And so what we'll have here is, again, I'll draw the docking proteins. Now, specifically, these docking proteins, they're called on the target membrane. So again, this is the plasma membrane of our neuron here, so plasma membrane, PM. Our docking protein is going to be consisting of something called T snares. And we won't get into detail about what proteins exactly encompass that T snare, but all you need to know is it's called a T snare. Similarly, you're going to have proteins on the vesicle membrane, which are called V snare. So you can kind of tell V snare, vesicular snare proteins, T snare, target membrane snare proteins. These are really important for understanding exactly how, uh, how, first of all, the vesicle, binds to the docking proteins, and also how the release of transmitters happens. And so one thing, I'm going to actually zoom in for the sake of drawing a little bit more clearly. But with these V snares and these T snares, normally without calcium, what you'll see is that they partially interact. They don't, they will sometimes interact, they will not, but they're not going to be bound tightly. And so what this is called docking. But this partial interaction, docking form allows the vesicle to just remain a little bit close to the docking proteins. And so I should also mention there is a protein here in this V snare complex that is called synaptotagmin. So here's the synaptotagmin. Synaptotagmin is a pretty cool protein because it has the ability to sense calcium. So it's a calcium sensor. So as we refer back to our sort of a zoomed out image here, so our calcium channels are right next to our docking protein here, our docking sites. So I'm going to draw that in here on the second image. So it's a little bit cut off, but here's our calcium channel. Again, you remember calcium channel, the calcium is going to flow into the cell because of diffusion. Calcium channel is going to flow, the calcium is going to flow in. Our synaptotagmin is going to be able to detect this local increase in calcium. And this is a huge step for triggering fusion of the actual membrane, or the fusion of the two membranes and fusion of the vesicles. The synaptotagmin is able to, which is part of that V snare complex, brings these two proteins very, very close together. So close together, in fact, that it's something called membrane zipping. And so what that means is that instead of having two membranes, what I'll draw here is actually you'll have it will look something like this, where the proteins will come together and the membranes will fuse together. So you won't see anything, any space between that. And that's called, again, as I mentioned, membrane zipping. And this zipping is pretty cool in that it happens so tightly that it prevents water from actually even getting through. So nothing can get through that zipping. And so as you remember before, so this is our vesicle. I'm gonna kind of overlay this a little bit. This is getting closer, then it's gonna fuse into the membrane. So again, a little bit more cleaner version on this side. Again, our vesicles and originally are right here, our neurotransmitters are right here. Our vesicles as fused with the plasma membrane, and now because you're kind of there's nothing preventing, there's no plasma membrane between this space, allows the neurotransmitters to release into the synaptic cleft. And again, once these neurotransmitters are released into the synaptic cleft, you can then bind to the receptors on the postsynaptic side. So again, this process actually happens at an extremely fast rate, which is about, I think, 0.5 to 1 millisecond. So you can imagine, I mean, all these things, they need to be primed and ready to go so that your neurotransmitters, your neurons can communicate with each other. And I always think it's always cool to see, these processes happening in action, or actually looking at the structure in real life. And so this is an electron micrograph. An electron micrograph is simply an image from an electron microscope. And we won't get into the details of how electron microscopy works in this video, but essentially what you can really appreciate in this video is that you can see the little bubbles here kind of look like essentially, our vesicles, and you'd be exactly correct if you guessed that. Our vesicles, you can see an outline. I'm going to just quickly outline here. Our presynaptic neuron, And then again, going back to our vesicles, they're going to have our neurotransmitters. And then you can see this little space here. I'll put it in green here. This little space, that's going to be our synaptic cleft. So, I've drawn it really, really big in uh, in this image here, but really you can really appreciate in this image that it's really, really small. Then all this sort of dense material here, this black stuff here, it's going to be all it's going to be all the receptors on the postsynaptic neurons. So I'll label that for you as well here. So again, something that, this process is very, very cool, happens very, very quickly. And really, I mean, something quite cool to consider too is you can actually begin to appreciate some other organelles here, which is, some of you probably guessed it already, is the mitochondria is here. So again, you're using a lot of proteins up, you're using a lot of things, a lot of activity happening at the synapse. So you want to make sure that you have enough energy. So our mitochondria can supply that energy. So yeah, that covers our presynaptic synaptic transmission portion. If you have any questions, feel free to leave them down below, and we'll cover some more topics in future videos. Thank you.